Well, good afternoon, everybody, and thank you all for coming to hear me speak to you about genomics and its time for translation into the clinic and its potential application to improve the health of patients worldwide. So I work at the William Harvey Research Institute in Queen Mary University of London, and I'm going to take you through some of the work that I've been doing at the Institute with my colleagues there. And then I'm going to focus in on a project that we have in the United Kingdom called the 100,000 Genomes Project. So our institute, as Professor Adana said, was founded by the Nobel laureate Sir John Vane, who's seen here receiving his Nobel medal from the King of Sweden. And he founded that institute 29 years ago. He discovered how aspirin worked. He also discovered prostacyclin, which paved the way for some treatments for a very serious lung condition, which is high pressure in the lung, called pulmonary hypertension. And he was also pivotal in the invention of drugs called angiotensin-converting enzyme inhibitors. And these are amongst the most widely used medicines worldwide to treat cardiovascular disease, heart failure, high blood pressure, and after heart attacks. So they're amongst the most commonly prescribed medicines today. Our institute is the largest pharmacological centre in the UK, and we benefit from having a very multidisciplinary environment focused on cardiovascular, inflammation, and endocrine research. We've grown since I became director from 140 people to 380 clinicians and scientists. And so we believe that we're at our best when we match clinicians and scientists, particularly when our focus is on biomedical health problems. Our institute is not full of British people. It has 46 different nations, and many of the groups and the centres are led from people from all over the world. And much as you've been doing the same sort of thing, in our institute 90% of the research has been rated independently by the UK government as world leading or internationally excellent. So I'm going to focus in on your and my genome. So as you sit there now, you are billionaires, because there's 3.3 billion letters in your genome. It may not be billionaires in money, but you certainly have 1 billion letters. And these make up your genetic code. And every 300 or so letters, there's a variation. This might be a single letter change, single nucleotide polymorphism. It might be an extra bit of DNA or a missing bit of DNA, an insertion, deletion polymorphism. Or it might be a copy number variant where bits of the DNA have been copied. And there are indeed other variations in the genome as well. These can tell us about susceptibility to disease. They can also, as I'm going to try and show you, prime therapeutic innovation, so give you and I the materials and the capability to develop treatments of the future. You could also use these variations to understand people's different response to drugs and why they might get adverse effects for some medicines. This is called pharmacogenetics. Through advances in technology over the last 20 years, we now have high throughput chips on which you can put 1 million or so of these variants. We now have massively parallel sequencing, which allows us to sequence efficiently and effectively in a short space of time a whole human genome. This is called next generation sequencing, and I know that you use it here at the Institute because I met somebody who is working with you on sequencing. Bear in mind that we actually have used most of the physiology or pharmacology available to us. We heard about a very interesting pathway a moment ago relating to icosanoids, lipoxygenase, and similar other areas. There may well be more drugs in those pathways, but actually, because in certain areas we're running out of medicines, we need to be able to generate new knowledge, and I think genomics give us a way into that. And it's important to set this in context. If it costs over $1 billion to develop a new medicine and 80% of them fail during their course of development, this is a serious problem for you and I, and we must expend all our endeavor to try and make sure in the future we can actually improve biomedical research. So we've organized our institute to work like this. So we believe that genomics can be used for target discovery and localization by integrating multiple different types of omics. So I've mentioned to you about whole genome sequencing. That's just one form of omics. We could look at the epigenome, We could look at the methylation patterns in the genome, your epigenome. We can look also at the proteome, the proteins in various body fluids, or the metabolome. The metabolome is metabolic signatures in body fluids. We can integrate data from all of this, and that can allow us to access causes of disease that then also identify pathways from which we can undergo target discovery. Once you've done that, what you need then to do is to have a group of people who are really top-class pharmacologists who integrate human lab studies. So in other words, outside the human body, but cell system studies on either inducible pluripotential stem cells or indeed on human cells in cell assays, and integrate those with experimental models, mouse transgenics, and cheminformatics, which India is extremely good at, and medicinal chemistry, which are also exceptional at. So this integration of all of these different modalities can allow you to develop new medicines. And because in our hospital, St. Bartholomew's Hospital, which was established in 1123, we now have centres that reach out across 6 million people, much like you have here in the population of Hyderabad. You have the opportunity to have a research window on your community and other communities like you around the world. And so we've integrated ourselves to do this, and increasingly this will allow us to bring new medicines into this space for patient benefit. So as Professor Adana said, I've been interested in blood pressure. Blood pressure is the #1 risk factor globally and is responsible for half of the 17.5 million deaths there are from cardiovascular disease each year. It's an extremely serious risk factor, and it's growing. A few years ago there were 1 billion people on Earth with high blood pressure. In 2025, just a few years from now, there will be 1.5 billion people with hypertension. So this is a global epidemic growing, and we've run out of pharmacology to exploit. And so looking at the genome may give us insights, you and I, that we can develop and then bring new medicines forward into the clinic. So I had the privilege of working in 2005 to 7 as part of the Wellcome Trust Case Control Consortium, whereas Professor Adana said I led the hypertension aspect of this. And this program compared 2,000 cases from 7 common diseases with 3,000 shared controls. And it used a genome-wide approach, looking at individual variants, so-called single nucleotide polymorphisms or SNPs, spread on a gene chip that a bit similar to a silicon chip. And that allows you to type many genome-wide variants that give you a picture of genome-wide variation. And from that, there were associations for some diseases, but sadly, the disease I was interested in, hypertension, had no significant findings whatsoever. So this got us thinking, what do we need to do? And there were several things that this told us. One is that we needed very large studies to make any inroads into the genetic basis of high blood pressure, and that possibly we needed slightly different approaches. So in this paper shown below, which we published in Nature from the International Consortium for Blood Pressure Genome-Wide Studies, we integrated the endeavor of 354 scientists from 234 institutions in 24 countries to study 200,000 people and discover over 29 genes for blood pressure. And these were amongst the first real breakthroughs. And that told us something about the genetics of blood pressure. The blood pressure is measured in millimeters of mercury, and these individual variants have very modest effects, small effects on these variants, on the blood pressure. Nonetheless, they do identify some pathways that we can exploit, and I'll share those with you. So what is the status currently of blood pressure genome-wide studies? Well, we have identified initially 34 loci. We've now extended that with other approaches, other types of gene chips, for a further 15. And in fact, in total today, there are 77 gene loci in the public domain. About 3% of the variance of blood pressure is explained by these. And as I said, they have modest effects. A millimeter of mercury on the systolic or 0.5 millimeter on the diastolic. And to give you an idea of what this means, although this is very modest effects, small effects on the blood pressure level, if I change the population of Hyderabad's blood pressure by just 2 millimeters of mercury, I can significantly change their risk of stroke and their risk of coronary disease. So if I increase their blood pressure, I can significantly alter this. And so you can see in aggregate, although these only explain a small proportion of the variance of blood pressure, they actually do represent some important clues to new physiology and potentially new pharmacology. So how we've taken this forward is to look at one particular pathway that emerged from this. Now, this pathway was known to be possibly implicated in blood pressure, but it wasn't known to be causatively implicated, and we now know that to be the case. And actually, in some measure, this pathway had been sort of looked at and set aside. In other words, people felt there wasn't anything there to be targeted, and so they didn't continue to research it and exploit it. We sometimes do this in medicine and in other branches of science. We start to look into something, we decide that maybe it's not a good idea, so we stop, and then 20 years later someone else picks up where you left off, and suddenly you discover something amazing. And in this case, this pathway, which is in your and my bodies, which causes vasodilatation, which produces a gas called nitric oxide, which opens up blood vessels, and the natriuretic peptides, this family of which cause salt and water loss in the kidney, these have been previously looked at as potential therapeutic modulators of blood pressure. But because the nitrates that were used at that time didn't work very well, and these proved difficult to target, it was abandoned. But interestingly, because we found not all of these elements now are associated with blood pressure elevation, including this here, which is a signaling protein, guanylyl cyclase, and subunits are associated with blood pressure level, what we were able to do is to identify medicines not being used to treat hypertension, but to be used to treat high blood pressure in the lung, and to consider working with Bayer to repurpose these for use in blood pressure lowering. Now what I'll show you is some medicines that we are now developing right now from this research. So this is my colleague Amrita Ahluwalia, with whom I work very closely. And what she has shown is that if you drink 250 mils of beetroot once a day, you can lower your blood pressure about the same amount as a standard dose of an antihypertensive medicine. Now, you might say, well, I'm not sure I'm a fan of beetroot. It makes your urine go red, and sometimes you get a bit of a bowel disturbance. But actually, if you live in a country where you don't earn very much money, and you have to make a choice between feeding your family or buying the medicines to treat your condition, what if you could treat your blood pressure and feed the family? So whilst beetroot juice might not be everybody's favorite food stuff, there are other foods, green leafy vegetables, in many countries of the world that also fix nitrogen. And what they have in them is inorganic nitrite. So this is some beetroot juice. You swallow the beetroot juice, it goes into the stomach, quite a lot of it goes out in your urine, but not all of it. And then the rest of it goes up to your salivary glands up here in your mouth. And it's concentrated there and then secreted onto your tongue. And in the back of your tongue live some anaerobic bacteria, so some bacteria, and they metabolize the nitrate to nitrite. When the nitrite is swallowed again in your stomach, it actually becomes nitric oxide, and this vasodilates you and lowers blood pressure. And what Professor Ahluwalia has shown is this can lower your blood pressure in the clinic and on ambulatory measurement by the same amount as a blood pressure lowering medicine. And this is now being used by patients in the UK as an adjunct to their antihypertensive therapy. Of course, we could take the nitrate out of this beetroot and make it into a tablet. That would be an option, but we haven't done that so far. So I'm going to just share with you some other work that my colleague Professor Adrian Hobbs has done, who I've been privileged to work with. And this is looking at the lining of your blood vessels. This is called the endothelium. This is the layer of cells that line your blood vessels. And they're stuck onto the outer side of smooth muscle. And this smooth muscle causes regulation of the tone, or how constricted your blood vessels are. And so what Adrian has done is he's looked at this molecule here, which is C natriuretic peptide. There's A natriuretic peptide, atrial natriuretic peptide, and B natriuretic peptide. But this one is produced inside the endothelium, and it acts on the smooth muscle through this receptor, and it causes vasorelaxation. There's also another receptor that it binds over here, which is called NPRC. This receptor, the gene encoding this, is associated with blood pressure elevation. And actually, we've recently found, and Professor Hobbs demonstrated this amongst others, that this receptor was thought to clear CNP, but it's now known to signal through a signaling mechanism and increase inwardly flowing or outwardly flowing potassium current, as a result hyperpolarizing the smooth muscle cell and again causing vasorelaxation. We can show this pathway is actually very, very effective by looking at a knockout mouse model. This is where we've disrupted the gene. But this is not a global knockout where you knock out the gene in every part of the animal. This is a tissue-specific knockout where we've knocked out the gene just in the endothelium, the lining of the blood vessel. Remember, I said to you a moment ago that that was where CNP was generated, and that's where it causes the vasorelaxation. So what Professor Hobbs did was he generated a knockout mouse model, and here's the aggregate data from all the mice, and you can see that if you have your wild type, your gene intact, you have a lower blood pressure over a 24-hour period than the knockout mouse where the gene has been disrupted but just in the blood vessel lining. Here's the blood pressure. So this is the mean 24-hour blood pressure. As you can see, much higher than the wild type with preserved CNP. So this suggests that you and I exist in a state of balanced vasodilatation, because this would be your normal state. This is the heterozygote, so you see almost a gene dose effect of the endothelial CNP gene. So this led Professor Hobbs to think, could we develop a medicine from this? And so he's developed a CNP mimetic, so C natriuretic peptide mimetic, that causes vasodilatation, and this is a dose response to increasing doses given IV. Now this molecule is not very soluble, but if we can make it soluble and make it into a tablet, it could be a new drug for high blood pressure. So this is taking genomics and using it to prime therapeutic innovation. Let's look at how genomics can help us with drug response. Here's a paper that I published towards the end of last year with my colleagues seen here in Nature Communications. And this is an analysis of about 40,000 people who are in statin trials or who've received statins. Statins are cholesterol-lowering medicines that were originally generated from yeasts and actually very powerfully lower cholesterol. However, what we found in this study was that by doing a genome-wide association study, we found that we could identify a number of new loci which explained 5% of the variation in patient response to cholesterol lowering. In other words, some patients given the same medicine had a different response to others, and as a result could be missing out on some of the benefits of the medicine. Now this is in its early days, but nonetheless this could pave the way for a series of gene tests. So what we've been doing at the William Harvey is to pull this together, and we're using the advantage of our rebuilding of this hospital here. This is a 60,000 meter squared new hospital which serves 6 million people for heart and cancer care, and this is the inside of that hospital. And what we're doing is inside there we're consenting all these patients who participate here to join us in studies that we can do, and to be able to recall them up to 4 times a year to offer them the benefits of our research. And this may allow us to bring genetics closer to the clinic, it may allow us to develop new medicines, and it may allow us to better predict their effect. I'm now going to move on to the 100,000 Whole Genomes project. And what I'm showing you here, because this may be difficult to work out what it is this is the opening ceremony of the London 2012 Olympics. And those 3 letters there, NHS, are the abbreviation of our National Health Service. So this is a healthcare system free at the point of delivery, and this is the only genomic transformation program for medicine in the world that's anchored in a healthcare system. So this is paid for by the National Health Service for patients in the National Health Service. And what we are doing is we've formed a company to deliver the program called Genomics England, and that's just there to move very quickly. And our mission is to sequence 100,000 whole genomes. So this is to read as much of your and my genetic code as we can, the entirety of the genetic code, and in rare disease, cancer, and in infectious agents. And so we're doing whole genome sequencing, not the coding regions or exomes. We're also not doing panel tests or other forms of sequencing. So this is next generation sequencing. In fact, at the moment, we use, like you do, HiSec 2500s. But in fact, we will shortly have one of the largest X10 facilities in the world, which is the latest form of machine. This leaves a legacy of infrastructure behind it, human capacity and capability to deliver genomic medicine in the clinic, and is aimed to generate health and wealth. And for the UK to be the first in the world, to be world leaders in the application of genomic medicine for healthcare, but to share that globally so that others can bring this into the healthcare system. So rare inherited diseases, you may be less familiar with this. Although rare inherited diseases are by definition rare and sometimes affect only 5 in 10,000 people, because there are so many of them, they affect about 5% of the UK population. That's about 3 million people in the UK. And this is because there are 7,000 rare disorders. For only half of those do we know the genomic diagnosis. So for only half of those disorders could I tell you what the genomic cause is. So what happens to these young people, and they're usually children or young adults, sometimes later onset, but mostly in early life. They go through life with a label, a name, a syndrome, but they don't actually know why they've got that disease. And the mothers and fathers of those children know if you and I can't tell them what the cause of their child's disease is, then we have little chance of understanding the biology that will allow you and I to develop a treatment for their child. And in some of these cases, it will be impossible to develop a treatment. But even in those settings, the mums and dads tell us consistently what they want to know from us is why their child or their loved one is like this. They want a diagnosis, and even that for them is something really big and important. So we know that early knowledge of the causes of this can avoid disability, but we also know that these diseases are usually disabling, often very costly, and the natural history of these patients is to move back and forth in the healthcare system. Whole genome sequencing from data we already have increases discovery for these patients and potentially diagnosis in about 25% over existing other tests. We're working at the moment on 110 different disorders. We're doing detailed characterization, and we're also getting all of the diagnostic tests that relate to these patients into our data center. We're anchoring the categorization of the clinical features these patients have in internationally accepted disease classifications. We're also measuring disease progression, and where clinicians think they've observed an additional characteristic that's not listed anywhere, we're including that characteristic too, because having that characteristic may help you and I to segment the disease and work out the different genetic causes. Because sometimes when we give these diseases a name, we give them a name because the patients with the disorder look similar. They may not be identical, but they look similar, they've got similar clinical features. But actually, when we get the genomic diagnosis, we find that in many cases, these diseases are not one single thing. There are several different things buried in the clinical features. So we're also building a national registry and linking the data to primary care every time you go to the hospital or if you die, to the outcome. Now, because these diseases are very, very rare indeed, sometimes it requires us to link people across the world. And this is a system called Decipher, which we're going to embed within our data center. And this is linking 1,800 clinicians across the globe. And these children and their parents have all consented to you seeing them and to the combination of their data anonymously, so we don't actually know who they are. And that anonymous combination for very ultra-rare disorders has allowed us to identify a cause for this disorder of mental retardation, speech delay, and epilepsy. So this is what we need to do to solve these diseases. Why is that important? Well, not for every single patient, but for some patients, it could allow us to develop medicines. Let me give you an example. So this is Orphanet, which is a consortium across Europe, and it's looking at diagnostic tests with the ambition of having 6,000 diagnostic tests for rare disease by 2020 and 200 medicines in development by 2020. Now, because these diseases are rare, until recently the pharmaceutical industry was less interested in developing medicines for these disorders. But now, thanks to different ways in which we pay for the medicines, they now are enthusiastic. And you can see by 2013 there were 64 drugs in development for rare diseases, where perhaps 5 or 10 years ago there would have been very few. So here's an example of just such a medicine. This is an enzyme that manages your cholesterol, PCSK9. A gain-of-function mutation in familial high cholesterol, 1 in 500, so slightly more people than could be in this lecture theatre. It was absolutely full, and you would have at least one person in this room with familial high cholesterol. Okay, so that's how common this is. It's a rare disease, but it's still quite common. And so this gene, gain-of-function mutation, causes a high level of cholesterol and increased coronary disease. By corollary, a loss-of-function mutation in Africans causes a big reduction in cholesterol, and this is the cholesterol that furs your arteries, and an 88% reduction in coronary disease. So this was discovered in 2005. By 2012, monoclonal antibodies targeting this enzyme were in phase 1 and 2 studies and have proven safe and effective, reducing cholesterol dramatically. Now, in my clinical research centre at the William Harvey Research Institute in London, we are doing trials on patients with coronary disease who've not responded to traditional cholesterol-lowering medicines or who can't tolerate those medicines. So here's an example of a finding in a rare disease helping us to prime a drug that may be of value for more common disorders with a similar characteristic. This is cystic fibrosis, one of the commonest genetic disorders there is. On the left is a chest X-ray showing the changes in the lung that occur in cystic fibrosis with overinflation and very big lung damage. Now, this has changed dramatically the life expectancy of these people, so they now live into their 30s and sometimes into their 40s. They get recurrent infections, their pancreas doesn't work very well, so they have to take enzymes. They have to have physio several times a day to remove the clogged up secretions, and they're usually on antibiotics. And so eventually the lung damage is so great they need a heart-lung transplant, which very few of them can get. Now, in November 2014, a company called Vertex filed with the Food and Drug Administration an application to launch this combination therapy, which opens the defective chloride channel, makes the secretions thinner, and can improve these patients' lung function. And even if this first medicine is not the answer, it has begun the process to get to the answer for these patients. What if we could change the outcome for these people so they don't have to die in their 30s? In cancer, the problem is more severe in many ways, because cancer is in essence, within the tumour, a disease of disordered genomes. There's over 200 driver mutations known. These tell us about drug targets. There's heterogeneity in the tumour, but you can use this information to stratify healthcare. In other words, you can use genotype to tell you what medicine to select, and I'll give you a couple of examples. We're working on lung, breast, colon, prostate, and ovary, the cancers from those organs, and also chronic lymphocytic leukemia, and we will work on these cancers here. We're also going to be charting the evolution of the cancer, and we will sequentially biopsy the cancer. So if someone's cancer comes back, we'll take another piece of the tumour and sequence it again. That's because sometimes the first treatment will remove the first set of driver mutations. This allows another set of driver mutations to become activated and to drive the tumour. This may also enable stratified healthcare, and we've linked to international consortia working on this. So on the left, you'll guess from the previous picture that this is a seriously abnormal lung. So this air is black on an X-ray. This person's should look like this. In fact, even this is still abnormal. This is the same person's X-ray, but air looks black on an X-ray. This person's got huge infiltration of cancer throughout the lungs, and as a result is unable to do anything, more or less. They're chair-bound, severely breathless, couldn't do any of the things you do normally, really, without being extremely breathless. But if I genotype their epidermal growth factor, I can gene, and I find a mutation, I can give them this medicine, gefetinib, that was going to end up on the shelf of a drug company. And that gefetinib changes this person's X-ray to that person's X-ray. This is not a cure. It's not going to remove the cancer. They will still eventually, sadly, die of the cancer. But what you've done is you've taken an inevitable short-term death and made that life expectancy longer. But more importantly than that, this person's not nearly as breathless. In fact, they have largely normal breathing. They can move around. They can probably even go to the shops unassisted. So this person now has their life back for a period of time. Here's another example. This is a PET CT scan. You probably mightn't be familiar with looking at these, but you'll be able to work out that's a human there. And if I tell you that the black is melanoma, you can see that this person has got melanoma, a cancer of the skin, all over them. This is the person before and after they receive a medicine that inhibits BRAF. This mutation is a driver mutation to response to BRAF inhibitors, and you can see that the cancer has largely disappeared from the person. Here's their skin full of lumps. This is them 6 weeks after a BRAF inhibitor. But that isn't the answer, because 23 weeks later they look like this. That's back, and it's back with a vengeance. What if you and I could sequentially biopsy this cancer, work out the evolutionary history of the architecture of the cancer, and develop better chemotherapies that we give at the outset so that this doesn't have to happen like this, and at the very least the cancer can become a chronic disorder, much as we've achieved for others. So in cancer, we're going to carefully characterize the patients, include their diagnosis and imaging. There is a problem in cancer in that the molecular pathology processes of dropping the tumor into a bucket of formalin is toxic to DNA and nucleic acids, as you probably will know. But even if you didn't, this is a real problem, because what the processing needs to do is to be nucleic acid friendly. So we're working on a system to allow us to do tumors at scale in a nucleic acid friendly way. We'll link all of the cancers to the specific cancer registries we have in our NHS, and also the chemotherapy and radiotherapy treatment and outcomes. So in pathogens, which may be closer to your interests here, we are looking at stratifying response and understanding adverse events and tracking outbreaks. So we're just beginning sequencing of 2,000 Mycobacterium TB isolates to look for resistance elements and to understand the epidemiology and origin of drug-resistant mycobacteria. Hepatitis C, now, this is the treatment of hepatitis C, effectiveness is predicted by genotype, and this is a potentially eradicable disease. There are medicines now that if we select them using genotype and given to the right person, we could get rid of this disease. Similarly, we could look at HIV. We can't necessarily get rid of it, but we could look at how we can use genomics there and in extreme response to infection. So we're advanced in implementing the program, but of course, you here now in the University of Hyderabad will know that whole genome sequencing is not the answer alone. You need more than that, and you need multi-omic samples to do transcriptomics, microRNAs, epigenetics, proteomics. You need to be able to look if there's cell-free circulating tumor DNA in the circulation. You need the sequential biopsies and other things too. So we're establishing a repository for that. And so we've just established 11 genomic medicine centres in England. These are centres of excellence that will generate the samples that we will work on and enrol the patients with very broad consent. This allows us to recall the patients to participate up to 4 times in further research projects. We're creating a national biorepository, and we're just building a sequencing centre with £27 million from the Wellcome Trust. This will create a refreshable, identifiable clinical data set, a life course registry, if you will, of these patients. We'll enrich that with data from their electronic records in primary care, hospital episodes, the cancer registries, and their outcomes. And all of this will be linked to whole genome sequence. We will then return results to the National Health Service, but we'll also push a pseudonymized, in other words, we've removed the identifiers so that this is anonymous now, data set in refreshable bills across into a data center where people like you, clinicians and academics, those in training and capacity building, and indeed industry will work together to drive up the clinical interpretation of these data. So We've established a partnership with Illumina, which means we never pay more than anyone else anywhere in the world for the same volume of sequence. We benefit from their latest chemistries and technologies, and they're going to run the sequencing center. We're not going to run the sequencing center. All universities, I expect here in India, to have Illumina's grey boxes that they no longer use sitting in the corner in a lab somewhere that should probably be in a museum. These are expensive, they're too expensive, so what we've done is said to Illumina, because you change your kit all the time, why don't you just provide the kit at your cost? And they're doing that, and this is excellent. This is a real partnership, and we're building this sequencing center, the ground floor of which here is our NHS Genomic Medicine Sequencing Center. I've mentioned we've created these NHS centers and the data center you saw a moment ago. But importantly for you, alongside this are 700 person years of master's, PhD and short course training to build the human capacity to deliver genomic medicine in the healthcare system. The other thing that we're doing is we're trying to break the cycle of implementation in a healthcare system. So if you and I come together and we form a scientific collaboration, we'll generate results and we'll publish those results. If we're working in medicine, we'll then try and get them implemented into healthcare. But this can take 17 years from research reports that have been evaluated recently. We don't have that time. So what we're doing is forming a coalition of the National Health Service clinical teams, academics and trainees, so in other words, people just like you, to work on this data from the outset, to drive up its interpretation, to evaluate its diagnostic potential and its therapeutic innovation potential, and allow us to implement this in the healthcare system with exactly the champions you need inside here to do that, and to do all of it by the end of 2017. So this is extremely ambitious. Clearly, it's very difficult to do. What does a domain within this partnership look like? Well, for any rare disease grouping, say cardiovascular disease, you will have a multifunctional domain where there are multiple different skill sets that combine to form a multidisciplinary team to allow us to drive up the interpretation of the data. We also open to industry partnership. This means that we want to encourage a hive of activity from pharma SMEs, diagnostic and therapeutic innovation, the sort of thing you have here in Hyderabad, in Genome Valley, and indeed in your computer science, which is revered around the world. So we're working with some international studies in collaboration, but there is no healthcare transformation program linked directly to an NHS-like system anywhere in the world. This is extremely challenging. It needs a coalition of a large number of intellects, including international collaborators, which could well be you. And we're going to support all of that with education to create a legacy of centres that can do this and the infrastructure to be able to bring it to the health service routinely. And the goal is new diagnostics and maybe, just maybe, new therapies for some of the people who suffer the most appalling diseases that face humankind. And we've not a real challenge to do this by the end of 2017. Here are some of the people that work with us on this project, and this is my team here. So you can see we're not spending the money on me, we're spending the money on the project, because there's not very many people. I went to visit one of your labs this morning. There were as many people in that lab as are in my science team, facing 100,000 whole genomes. And this is the groups I've worked with to generate the data on blood pressure I shared with you earlier. And these are the people I work with, along with another 350 or so around the world, to generate the blood pressure gene discoveries. It's been a real honor to join you here at the University of Hyderabad. I would like to thank the Vice-Chancellor and you, Professor Radana, for your extremely warm welcome, and each of you for coming here today. And hopefully you will see how we could do something here and give thought to how we might be able to bring this type of activity into clinical practice here in India. Thank you very much indeed.